Life on Earth did not adapt to the planet. Life remade the planet for itself. Every atom of oxygen in the atmosphere is of biological origin. Every molecule of coal and oil is of biological origin. Half of all sedimentary rock is of biological origin. Soil is of biological origin. If you removed everything that life has created, Earth would once again become a dead rock with a poisonous atmosphere. We do not live on a planet. We live inside the largest biological project in the history of the solar system. And new research published in 2025 explains for the first time why this project almost failed 2 billion years ago and what saved it. Before we go any further, please subscribe to this channel. Your support really helps us to continue our work. Thank you. There is a story about the Earth that almost everyone has heard, and almost everyone has backwards. The story goes like this. The Earth formed. Conditions gradually became suitable for life. Life appeared, and life then flourished in the environment that the planet had prepared for it. This story is wrong in almost every particular that matters. The Earth that formed 4.5 billion years ago was not a place that was waiting to become suitable for life. It was a place that was, by any standard that biology recognizes, actively hostile to the kind of life we know. And the transformation of that hostile place into the world we inhabit was not something the Earth did on its own. It was something life did to the Earth. The planet we live on is not the original planet. It is a biological product, a world that has been chemically, geologically, and atmospherically rewritten by living organisms over 4 billion years of continuous biological engineering. The original Earth, the Earth as it existed in the Hadean Eon from approximately 4.5 to 4 billion years ago, was a world that geologists have spent decades trying to reconstruct from the fragmentary evidence available, because the Hadean rock record is almost entirely gone, destroyed by the relentless geological activity of the early planet. What we know about the Hadean comes primarily from zircon crystals, the most geologically resistant minerals on Earth, found in ancient Australian formations and dated to approximately 4.4 billion years ago. These zircons were preserved within younger rocks, while the original igneous crust that contained them was destroyed and recycled, and they preserve within their crystal structure the chemical signatures of the conditions under which they formed. What the zircons tell us is that the Hadean Earth had liquid water. The oxygen isotope ratios in the zircons are consistent with crystallization in the presence of liquid water, suggesting that the surface was cool enough for oceans to exist within a few 100 million years of the planet's formation. This was once thought to be impossible. The conventional picture of the Hadean was a hellish world of continuous bombardment and surface temperatures too high for liquid water, but the zircon evidence has revised that picture substantially. The early Earth may have been less continuously hellish than was assumed, with the liquid water oceans appearing relatively early. But liquid water does not mean habitable. The Hadean atmosphere was radically different from the modern atmosphere. It was dominated by carbon dioxide, water vapor, nitrogen, methane, and ammonia. The volcanic gases that the young Earth was outgassing from its interior at enormous rates, as the heat of accretion and radioactive decay drove continuous geological activity. There was essentially no free molecular oxygen. The early ocean was hot, reducing, and acidic, a chemical environment utterly unlike the modern ocean, with chemistry that would be lethal to the vast majority of organisms alive today. This is the starting point. This is the planet that life inherited and proceeded to transform. The distance between that planet and the planet we live on today is the measure of what 4 billion years of biological activity has accomplished. It is an enormous distance, arguably the largest single transformation in the history of any planet in the solar system that we know about. And it was accomplished not by geology alone, but by the accumulated metabolic activity of trillions upon trillions of individual organisms over geological time. The first life appeared in this hostile environment somewhere between 4 and 3.5 billion years ago. The oldest evidence for life comes from carbon isotope signatures in rocks approximately 3.5 to 4 billion years old, from the Pilbara region of Australia and the Isua Formation of Greenland, though the interpretation of the very oldest signatures is contested. What is not contested is that by approximately 3.5 billion years ago, life was present and metabolically active in the early ocean. These first organisms were not photosynthesizers. They were chemolith or autotrophs, organisms that extracted energy from inorganic chemical reactions rather than from sunlight. The first metabolisms on Earth were almost certainly based on the chemistry of hydrothermal vent systems, where the geological activity of the planet itself provided the chemical gradients. The differences in oxidation state and temperature between the hot, mineral-rich vent fluid and the cold, relatively oxidizing deep ocean water that early life exploited for energy. To understand what the first life actually looked like and how it functioned, you need to descend to the bottom of the Archean Ocean and find the hydrothermal vents. These are not merely geological features. They are, in the current best understanding of the origin of life, the cradle in which the chemistry of living systems first assembled itself into something that could replicate, metabolize, and evolve. The hydrothermal vents of the Archean Ocean were different from the famous black smoker vents of the modern deep ocean, which are hot, acidic, and driven by the heat of newly formed oceanic crust. The vents most commonly implicated in the origin of life are alkaline hydrothermal vents, structures formed when seawater percolates into the oceanic crust and reacts with iron-rich rocks in a process called serpentinization, emerging at the seafloor as warm, alkaline fluid rich in hydrogen, methane, and ammonia. The Lost City hydrothermal field on the Mid-Atlantic Ridge, discovered in 2000, is the best modern example of this type of vent system, and its towering carbonate chimneys, some reaching 60 meters in height, have become the template for thinking about the environment in which life first appeared. The walls of these chimneys are riddled with tiny pores and channels, each only a fraction of a millimeter in diameter, separated by thin mineral walls that create natural electrochemical gradients between the alkaline interior fluid and the more acidic exterior ocean. These gradients, differences in pH and in the concentration of hydrogen ions, are directly analogous to the electrochemical gradients that every living cell on Earth uses to generate energy. The membrane potential that drives the synthesis of ATP in your mitochondria is, at its most fundamental level, the same kind of proton gradient that the mineral walls of alkaline hydrothermal vents generate spontaneously. The hypothesis, developed by Mike Russell and Nick Lane among others, is that life did not invent this energy-generating mechanism from scratch. It inherited it from the geology of the vent. The first cells were, in a sense, mineral pores that learned to maintain their own gradient rather than depending on the rock to do it for them. Whether or not this specific hypothesis is correct in its details, and it remains an active area of research, the broader picture it suggests is revolutionary in its implications. Life did not appear on a planet and then begin to interact with it. Life appears to have emerged from the interaction between chemistry and geology at the precise interface where the two systems were already exchanging energy. The first organisms were not visitors to a geological world. They were products of it, extensions of the geochemical processes that were already running, but extensions that had acquired the ability to catalyze those processes, to concentrate them, and eventually to direct them. From the beginning, life and geology were not separate systems operating in parallel. They were one system with an increasingly biological character. The organism at the root of the evolutionary tree, the last universal common ancestor, LUCA, whose existence can be inferred from the features shared by all living things, was almost certainly an anaerobic, chemolithotrophic organism living in or near hydrothermal vents, using the hydrogen produced by serpentinization reactions as an energy source and CO2 as a carbon source. It was not simple in the biochemical sense. LUCA already possessed a sophisticated genetic code, ribosomal machinery for protein synthesis, and a complex array of metabolic enzymes. But it was simple in its ecological role. A single organism exploiting a single geochemical energy source in a single type of environment. The diversity of metabolic strategies that characterizes the modern microbial world, sulfate reduction, iron reduction, methanogenesis, acetogenesis, anoxygenic photosynthesis, oxygenic photosynthesis, aerobic respiration, was built up over the following hundreds of millions of years as evolutionary innovation opened new energy sources and new environments to biological exploitation. The methanogens, the archaea that produce methane by combining hydrogen and carbon dioxide, deserve more than passing mention as the first planetary engineers. The scale of methanogen activity in the Archean Ocean is difficult to visualize, but possible to quantify from the geological record. The carbon isotope signatures of Archean organic matter, the sulfur isotope records of Archean sediments, and the modeling of Archean climate under the faint young sun all converge on the same conclusion. The Archean atmosphere contained methane at concentrations orders of magnitude higher than today, and this methane was biologically produced. Estimates of Archean methane concentrations range from 100 to 1,000 parts per million, compared to the modern approximately 2 parts per million. And this methane haze would have given the Archean sky an orange color similar to the present-day atmosphere of Saturn's moon Titan. This methane haze was not merely a curiosity. It was the difference between a frozen planet and a habitable one. The faint young sun, 30% less luminous than today, provided insufficient energy to maintain liquid water on the Earth's surface without substantial greenhouse warming. The CO2 in the Archean atmosphere contributed some of this warming, but modeling studies consistently show that CO2 alone was probably insufficient to fully resolve the faint young sun problem. Methane, with its much more powerful greenhouse effect per molecule, makes up the difference. The methanogens were, without any intention or awareness, running a planetary climate control system, producing precisely the greenhouse gas in precisely the quantities needed to keep the ocean liquid and the planet habitable. If the methanogens had not evolved, or had evolved later, or had been less productive, the early Earth might have experienced runaway glaciation, from which recovery would have been difficult or impossible. The diversity and abundance of all subsequent life on Earth is contingent, in this sense, on the metabolic activity of the archaea that learned to make methane in the Archean Ocean. The first 3 billion years of life on Earth were overwhelmingly microbial, and this period is where the foundational rewriting of the planet occurred. Every major biogeochemical cycle, the carbon cycle, the nitrogen cycle, the sulfur cycle, the phosphorus cycle, the iron cycle, was established, modified, and ultimately dominated by microbial metabolic activity before the first animal ever existed. The microbial world built the biochemical infrastructure of the Earth system, the enzymes that fix nitrogen from the atmosphere, the metabolic pathways that oxidize sulfur and iron, the photosynthetic machinery that captures sunlight and uses it to reduce carbon. These biochemical inventions are the foundation on which all subsequent biological complexity rests. The animals and plants and fungi that have occupied the last 600 million years of Earth history are, in the most fundamental sense, the passengers on a biological ship that microbes built and still operate. The ocean of the Archean and early Proterozoic was a radically different chemical environment from the modern ocean. It was dominated by iron in its reduced ferrous form, which is soluble in water and was present throughout the water column at concentrations many orders of magnitude higher than in the modern ocean. It was essentially devoid of free sulfate. It was anoxic from surface to floor, and it was populated by a microbial community of staggering metabolic diversity: sulfate reducers, iron reducers, methanogens, acetogens, anoxygenic photosynthesizers, each exploiting a different chemical gradient, each producing waste products that became the energy source for another group, each weaving the chemical cycles of the early ocean into an increasingly complex web of biological interdependence. This is the world that the cyanobacteria were about to transform beyond recognition. The organisms that first populated these environments were metabolically diverse in ways that are only now being understood. Sulfur-reducing bacteria, iron-reducing bacteria, methanogens that combined hydrogen and carbon dioxide to produce methane, acetogens that produced acetate by similar pathways. The methanogens deserve particular attention because they were the first organisms to make a significant change to the Earth's atmosphere. Methane, the product of methanogen metabolism, is a potent greenhouse gas. Approximately 80 times more effective at trapping heat than carbon dioxide over a 20-year period. The early Earth had a significant methanogen problem in the most consequential geological sense. The young sun was approximately 25 to 30% less luminous than it is today, and without the warming effect of greenhouse gases, the early Earth should have been frozen solid under the faint young sun. The methane produced by the early biosphere, pumped into the atmosphere by trillions of methanogenic archaea in the early ocean, was a significant contributor to maintaining the warmth that allowed liquid water to persist. Life, in its first significant act of planetary engineering, helped keep the early Earth from freezing. The functional consequence is clear regardless of how we classify it. Life was changing the planetary climate within its first billion years. And this was only the beginning. The transition that defined the next chapter of the Earth's biological history is called the Great Oxidation Event, and it is arguably the most consequential single biological transition in the history of the planet. It occurred approximately 2.4 billion years ago, and it was caused by an organism so small that you could fit 1 billion of them in a teaspoon of seawater. The organism was cyanobacteria. The consequence was oxygen. Cyanobacteria evolved the ability to perform oxygenic photosynthesis, a metabolic pathway that uses sunlight to split water molecules, releasing oxygen as a byproduct and using the hydrogen to reduce carbon dioxide into organic carbon. The chemistry of this reaction is straightforward. Its consequences were not. Oxygen, free molecular oxygen O2, was a poison to virtually every organism then alive on Earth. The entire existing biosphere had evolved in the absence of free oxygen, and its metabolic machinery had no defenses against the reactive chemistry of an oxygen-rich environment. When the cyanobacteria began pumping oxygen into the ocean and eventually into the atmosphere, they were committing what is, in terms of the number of species killed, the largest mass extinction in the history of life on Earth. The research, published in November 2025 by Ratnayake, Tanaka, and Nakamura at Okayama University in the journal Communications, Earth and Environment, adds a crucial new dimension to this story. For decades, one of the central mysteries of the Great Oxidation Event has been the timing question. If cyanobacteria evolved oxygenic photosynthesis as early as 3.5 billion years ago, and the molecular clock evidence strongly suggests they did, why did free oxygen not accumulate in the atmosphere until approximately 2.4 billion years ago? What held oxygen back for more than 1 billion years? Previous hypotheses had focused on the capacity of the early ocean and atmosphere to absorb oxygen through chemical reactions. The idea that oxygen was being produced but immediately consumed by reactions with reduced iron, sulfide, and methane before it could accumulate. The Okayama study identifies 2 additional factors that had been previously overlooked: nickel and urea. High concentrations of nickel in the early ocean derived from the volcanic activity that was far more intense in the Archean than today, appear to have suppressed cyanobacterial growth by acting as a metabolic inhibitor. High concentrations of urea, derived from the breakdown of organic nitrogen compounds, similarly limited cyanobacterial proliferation. As volcanic activity gradually decreased through the late Archean and early Proterozoic, nickel concentrations in the ocean declined. As the chemistry of the early ocean shifted, urea concentrations stabilized. The cyanobacteria were released from their chemical suppression and underwent the explosive population growth that produced the oxygen pulse of the Great Oxidation Event. The Great Oxidation Event was not a sudden switch from an oxygen-free to an oxygen-rich world. It was a transition that unfolded over approximately 300 million years, from roughly 2.4 to 2.1 billion years ago, and it was punctuated by episodes of much more extreme oxygenation, followed by periods of partial reversal. The consequences for the biosphere were catastrophic for the organisms that could not adapt. Anaerobic organisms, those that had evolved in the absence of oxygen and for which oxygen was toxic, were driven into refugia. The oxygen-poor sediments, the deep ocean below the oxygen minimum zone, the anoxic interiors of microbial mats. Many lineages went extinct entirely. The Great Oxidation Event was, from the perspective of the anaerobic biosphere that had dominated the Earth for the previous billion years, a global catastrophe caused by the metabolic activity of a single group of organisms. But the oxygen that was killing the anaerobic world was simultaneously creating the conditions for something far more complex. Oxygen in the atmosphere is not merely a chemical, it is an energy currency. The amount of energy that can be extracted from organic molecules through aerobic respiration is approximately 18 times greater than the amount that can be extracted through anaerobic fermentation. An organism with access to oxygen can sustain a metabolic rate, a level of complexity, and a body size that is simply impossible without it. The Great Oxidation Event was not only the largest mass extinction in Earth history, it was also the prerequisite for every complex organism that has ever lived. Every animal, every plant, every fungus, every organism visible to the naked eye. The cyanobacteria poisoned the world, and the world they poisoned became the world that could produce us. The oxygen revolution was not the only consequence of the Great Oxidation Event. The transition to an oxidizing atmosphere had profound effects on the chemistry of the Earth's surface, the composition of the ocean, and the nature of the rock record. Iron, which had previously existed in the reduced ferrous form that is soluble in water and therefore present throughout the Archean Ocean, was oxidized to the insoluble ferric form and precipitated out of the ocean onto the seafloor in the massive deposits of banded iron formation that characterize the Proterozoic geological record. The great banded iron formations of Australia, South Africa, Canada, and Brazil, rocks that are now mined as iron ore for steel production, are a direct product of the Great Oxidation Event. They are the rust of a planet's first breath. Every piece of steel produced from these formations is a physical artifact of biological activity that occurred 2 billion years ago. The elevated methane levels of the late Archean, maintained by the biosphere's methanogen community, were simultaneously disrupted by the rising oxygen. Oxygen is chemically incompatible with methane under the conditions of Earth's atmosphere. The two react to produce carbon dioxide and water. As oxygen levels rose and methane levels fell, the greenhouse effect that had been maintaining the Earth's warmth was suddenly reduced. The planet cooled. The cooling was not gradual. It was severe enough to trigger the first of Earth's great glaciations, the Huronian glaciation, which began approximately 2.4 billion years ago and extended in some episodes to glaciers reaching the equator. This is the first of what geologists call snowball Earth events, episodes of extreme global glaciation in which the entire surface of the planet, including the tropical oceans, was covered in ice. Life, in producing oxygen, had inadvertently destroyed the greenhouse warming that had kept the planet warm and nearly frozen itself into extinction. The snowball Earth events are among the most dramatic episodes in Earth history, and they are, in their origin and their termination, deeply biological stories. The glaciations were terminated not by astronomical changes, but by volcanic outgassing. The continuous eruption of CO2 from volcanoes that persisted even when the surface was frozen could not be absorbed by the ice-covered ocean, and over millions of years it accumulated to levels sufficient to produce a greenhouse warming that melted the ice. When the ice melted, the sudden exposure of enormous quantities of meltwater rich in nutrients from the glacially weathered rock triggered explosive blooms of photosynthetic life, biological explosions that themselves modified the atmospheric chemistry, produced more oxygen, and may have contributed to the biological diversification that followed each glaciation event. The period between approximately 1.8 and 800 million years ago, roughly a billion years of Earth history, has traditionally been described as the boring billion, a time of apparent biological and geological stagnation in which the Earth changed little and life diversified slowly. The research published in October 2025 by Muller and colleagues at the University of Sydney and the University of Adelaide in the journal Earth and Planetary Science Letters challenges this characterization in important ways. The breakup of the supercontinent Nuna, approximately 1.5 billion years ago, the study shows, was not geologically or biologically uneventful. The fragmentation of Nuna expanded the area of shallow continental shelves, the nutrient-rich, sunlit marine environments most suitable for complex life. It reduced volcanic CO2 emissions as the supercontinent's interior was replaced by new ocean crust. It cooled the climate, and it created the oxygenated shallow water habitats that were the necessary precondition for the origin of eukaryotes, cells with nuclei, the ancestors of all complex life. The eukaryotic cell is a biological structure of extraordinary complexity and consequence. Its defining feature, the membrane-bound nucleus containing the chromosomal DNA, represents a fundamental reorganization of the cell's genetic and metabolic machinery that enabled the evolution of sexual reproduction, complex gene regulation, and the elaboration of cell types that made multicellular organisms possible. The origin of the eukaryotic cell, the event that made all complex life possible, deserves to be told in the detail it earned. The story of the mitochondrial endosymbiosis, the event in which an archaeal cell engulfed a bacterial cell and initiated the evolutionary partnership that produced the eukaryotic cell, is one of the most extraordinary stories in the history of life, and it deserves to be told in more detail than the summary usually provided. The conventional account treats the endosymbiosis as a single event. A cell swallowed another cell and retained it as an internal organelle. But the reality was almost certainly more complex, more gradual, and more contingent. The archaeal host cell that gave rise to the eukaryotic lineage was a member of the Asgard archaea, a group of deep-branching archaea discovered in seafloor sediment samples from the Loki's Castle hydrothermal vent field on the Arctic mid-ocean ridge. First described in 2015. Living representatives of Asgard archaea have proved extraordinarily difficult to culture in the laboratory. But metagenomic analysis of their DNA has revealed that they possess many of the protein families previously thought to be exclusively eukaryotic. Proteins involved in cell membrane dynamics, cytoskeletal organization, and intracellular trafficking. This means that the archaeal host cell was already, in some sense, pre-adapted for eukaryotic complexity before the endosymbiosis occurred. The architecture of the eukaryotic cell was not built entirely from the bacterial symbiont. It was built by the combination of archaeal sophistication with bacterial energy production. The bacterial partner that became the mitochondrion was an alpha proteobacterium, a member of the same bacterial group that includes the modern rickettsia and anaplasma, intracellular parasites of eukaryotic cells that represent, in a sense, the reverse of the endosymbiotic relationship. Organisms that invade and exploit eukaryotic cells, rather than cooperating with them. The transition from a free-living alpha proteobacterium to an obligate intracellular symbiont to the fully integrated organelle we call the mitochondrion involved the progressive transfer of genetic information from the symbiont's genome to the host's genome, and the corresponding evolution of protein import machinery to deliver the products of those transferred genes back into the organelle. Modern mitochondria retain only a small fraction of the original bacterial genome. In humans, the mitochondrial genome encodes only 13 proteins, while the hundreds of other proteins required for mitochondrial function are encoded in the nuclear genome and imported into the mitochondrion after synthesis in the cytoplasm. This evolutionary fusion produced an organism of unprecedented metabolic capacity. The eukaryotic cell, powered by its mitochondrial engines running aerobic respiration, could generate approximately 18 times more ATP per molecule of glucose than any anaerobic organism. This metabolic surplus funded the evolutionary elaboration of cellular complexity. The nucleus, the cytoskeleton, the endoplasmic reticulum, the Golgi apparatus, the complex gene regulation that made the evolution of different cell types possible. The energy freed by aerobic respiration was, in the most literal sense, the energy that paid for the evolution of complex life. Size, complexity, and multicellularity all require metabolic investment, and the mitochondrial endosymbiosis made that investment affordable for the first time. The chloroplast, the photosynthetic organelle of plants and algae, has a parallel origin. It is a cyanobacterium that was engulfed by a eukaryotic cell approximately 1.5 billion years ago and retained as an endosymbiont, eventually becoming the integrated organelle responsible for all plant photosynthesis. The cyanobacteria that had poisoned the Archean biosphere with their oxygen were engulfed by the eukaryotic cells that had evolved to tolerate and exploit that oxygen, and became the photosynthetic engines of the plant kingdom. This is biological recycling at its most extraordinary. The organisms responsible for the great oxidation event were incorporated into the cells that the event had made possible, and became the primary photosynthetic machinery of all complex plant life. Every green thing you have ever seen, every blade of grass, every tree, every vegetable, is photosynthesizing through organelles that are the evolutionary descendants of the same cyanobacteria that nearly destroyed the early biosphere 2 billion years ago. The Ediacaran biota, the strange, soft-bodied macroscopic organisms that appeared approximately 600 million years ago, following the final snowball Earth glaciations of the Cryogenian, represent the first experiment in macroscopic biological complexity, and they are worth pausing on because they illustrate how radically different the biological world of even the recent geological past was from anything alive today. Dickinsonia, one of the most studied Ediacaran organisms, was a flat, oval, rib-like animal that could reach lengths of nearly 1.5 meters. It had no mouth, no gut, no visible sensory organs, no appendages. It appears to have moved slowly across microbial mats on the Ediacaran seafloor, absorbing nutrients through its ventral surface in the manner of a living carpet rather than an animal in any sense that modern biology would recognize. Whether Dickinsonia was an animal, a relative of modern animals, or something else entirely, a biological experiment that left no descendants, is still actively debated. What is certain is that it was macroscopic, multicellular, and dependent on the oxygen that the cyanobacteria had been producing for 2 billion years. The Ediacaran world was inhabited by dozens of such organisms. Charnia with its frond-like structure, Tribrachidium with its threefold symmetry found nowhere in the modern animal kingdom, Kimberella, which shows some evidence of bilateral symmetry and directed movement. These organisms lived in a world that was, by the standard of the previous 3 billion years, biologically revolutionary, macroscopic, multicellular, and aerobic. But it was a world without predators. No organism in the Ediacaran appears to have been capable of actively attacking and consuming another macroscopic organism. It was, in the phrase of the paleontologist Andrew Knoll, a world of peaceful giants, populated by organisms that grazed on microbial mats and absorbed dissolved organic carbon from the water, lacking the ecological arms race between predator and prey that would transform the biological world in the event that followed. The Cambrian explosion began approximately 538 million years ago. Within approximately 20 million years, a span of time that is geological instant, representatives of most major animal body plans appeared in the fossil record. trilobites with their compound eyes and segmented bodies, Anomalocaris with its grasping appendages and circular tooth-ring mouth, Picaia, the earliest known chordate, a small worm-like animal with a notochord that is the ancestor of every vertebrate alive today, Hallucigenia, whose bizarre appearance, spines on one side, tentacles on the other, confused paleontologists for decades about which end was the head and which was the tail. The organisms of the Burgess Shale of British Columbia and the Chengjiang biota of Yunnan province in China are among the most intensively studied fossils in the world because they preserve, in extraordinary soft tissue detail, a snapshot of the biological diversity that appeared within a geological instant at the base of the Cambrian. The Cambrian explosion was not random. It was the product of the convergence of multiple enabling conditions that had been building for hundreds of millions of years. Atmospheric oxygen had risen to levels that could support the elevated metabolic rates of active predatory animals. The shallow continental seas, expanded by the breakup of the supercontinent Rodinia, provided vast areas of sunlit, nutrient-rich water for the explosion of primary productivity that the explosion's food web required. The evolution of biomineralization, the ability to build hard shells and skeletons from calcium carbonate and calcium phosphate, provided both defensive armor against predators and a structural framework for larger body sizes. And the ecological innovation of predation, one organism actively hunting and consuming another macroscopic organism, triggered the evolutionary arms race between predator and prey that drove the rapid diversification of body plans, sensory systems, locomotory mechanisms, and defensive structures that characterizes the Cambrian explosion. The eyes that appeared in the Cambrian, compound eyes in trilobites, simple eyes in other Cambrian animals, were not merely biological innovations. They were planetary ones. The evolution of vision created a selective pressure for speed, camouflage, signaling, and behavioral complexity that had no precedent in the pre-Cambrian world. Light, which had been driving photosynthesis for billions of years, was now also driving the evolution of the most complex nervous systems that life had yet produced. The information processing capacity of the Cambrian animal nervous system was a qualitative leap beyond anything the microbial world had achieved, and it set the trajectory for the subsequent evolution of neural complexity that eventually produced the human brain. The chain from cyanobacterium to human is unbroken, and every link in it runs through oxygen. The eukaryotes diversified slowly through the boring billion, constrained by the relatively low oxygen levels of the Proterozoic atmosphere and ocean, and by the limited nutrient availability of the deep-time ocean. The single-celled eukaryotes of this period, protists, algae and their relatives, left a fossil record that is sparse and difficult to interpret, but that documents the slow accumulation of biological complexity in the microbial world. Then, approximately 600 million years ago, something changed. The Ediacaran period, from approximately 635 to 538 million years ago, is named for the Ediacara Hills of South Australia, where the first fossils of large, complex, multicellular organisms were found in the 19th century. The Ediacaran fauna, soft-bodied organisms of uncertain biological affinity, some reaching lengths of a meter or more, represents the first appearance of macroscopic multicellular life in the fossil record. They are strange animals, flat, frond-like or disc-like forms with no clear anatomical organization into the organs and systems that characterize the Cambrian and later animals. Whether they were the ancestors of modern animal phyla or an evolutionary dead end unrelated to living animals is still debated. What is clear is that their appearance coincides with a period of elevated atmospheric oxygen, the Neoproterozoic oxygenation event, a second major pulse of atmospheric oxygenation that followed the breakup of the supercontinent Rodinia and the snowball Earth glaciations of the Cryogenian period. The Cambrian explosion, the geologically rapid appearance of representatives of most major animal body plans in the fossil record, approximately 538 million years ago, is the most dramatic single episode of biological diversification in the history of complex life, and it was made possible by the same biological infrastructure that had been building for the preceding 2 billion years. The oxygen levels of the Cambrian atmosphere and ocean were high enough to support the active predatory lifestyles and the complex internal organs of the newly diversifying animal phyla. The nutrient-rich sediments of the continental shelves, themselves a product of billions of years of biological weathering and the chemical transformation of the ocean by photosynthetic life, provided the food base for the explosion of herbivores and filter feeders that preceded the predator diversification. The Cambrian explosion was not a random event or a geological accident. It was the fruit of 2 billion years of biological preparation. The transformation of the land surface by life is perhaps the most dramatic visible example of biological planetary engineering, because the evidence is everywhere, in every handful of soil, in every forest, in every river that runs brown with dissolved organic material from the land it drains. The original land surface of the early Earth was bare rock and mineral. There was no soil in the biological sense, because soil is not simply ground-up rock. It is rock transformed by the metabolic activity of organisms. The development of terrestrial ecosystems over the past 500 million years has created, from bare rock, a planet-wide biological system that processes carbon, nitrogen, phosphorus, and water at planetary scale. The colonization of land by plants approximately 470 million years ago was not a single event, but a process that unfolded over tens of millions of years. From the first simple bryophyte-like land plants of the Ordovician, through the development of vascular plants and the appearance of the first forests in the Devonian. The research published by the Biosphere 2 team in September 2025 makes the mechanism of this transformation vivid. Plant roots inject carbon dioxide directly into the soil where they contact rock, driving the chemical weathering reactions that break down minerals and release nutrients. Before plants colonized the land, weathering was driven by purely physical and chemical processes: rain and frost and temperature cycling. After plants, weathering was driven by biological activity, and the rate of weathering increased dramatically. The consequence was a massive drawdown of atmospheric CO2. Carbon dioxide was pulled from the atmosphere by the weathering reactions that plant roots were driving, and the carbon was locked in soil, sediment, and eventually rock. Global temperatures fell. The Devonian and Carboniferous were in part biologically cooled by the emergence of land plants. The Carboniferous coal forests are the most visible record of this biological cooling, and they illustrate another dimension of the biological transformation of the planet. The trees of the Carboniferous, the lycopsid giants, the giant horsetails, the first seed plants, were producing enormous quantities of woody biomass that accumulated in the swampy coal forests faster than it could be decomposed. The reason for this accumulation was biological. The microorganisms capable of decomposing lignin, the structural polymer that gives wood its rigidity, had not yet evolved. The first true lignin-degrading fungi appear in the fossil record only in the late Carboniferous or early Permian, and their evolution is directly correlated with the end of the coal-forming epoch. Before the lignin degraders evolved, the organic carbon in fallen wood accumulated as peat and was eventually transformed by geological processes into coal. The coal seams that powered the Industrial Revolution and that continue to power a substantial fraction of global electricity generation today are the compressed bodies of 300-million-year-old trees that fell into swamps and were not consumed by decomposers because the decomposers had not yet evolved. The energy that drove the industrialization of human civilization is biological in origin, stored 300 million years ago by organisms that could not have imagined the use to which their bodies would eventually be put. The same biological origin applies to petroleum. Oil is not a mineral in the geological sense. It is not produced by inorganic processes under high temperature and pressure. It is the transformed product of marine organic matter, the bodies and biological molecules of phytoplankton, bacteria, and other marine organisms that sank to the seafloor, were buried by sediment, and were cooked over geological time into the hydrocarbon mixtures we call petroleum. Every barrel of oil produced from any oil field in the world is a sample of ancient biological material. The carbon in the CO2 that is released when that oil is burned was originally fixed from the atmosphere by photosynthetic organisms tens to hundreds of millions of years ago. The combustion of fossil fuels is, in the most literal chemical sense, the reversal of the biological processes that removed carbon from the atmosphere and stored it in geological formations over geological time. The limestone that forms the white cliffs of Dover, the coral reefs of the tropical Pacific, the Jurassic limestone formations of the European Alps, the Cretaceous chalk formations of the Great Plains of North America. All of these are biological in origin. Limestone is calcium carbonate, and the calcium carbonate in limestone was deposited by the organisms, corals, foraminifera, coccolithophores, brachiopods, mollusks, that built shells and skeletons from the calcium and carbonate dissolved in seawater. When these organisms died, their shells accumulated on the seafloor in quantities that over geological time were compressed and cemented into the carbonate rock sequences that make up a substantial fraction of the Earth's sedimentary record. The white cliffs of Dover are the compressed shells of coccolithophore algae that lived in the Cretaceous Ocean approximately 66 to 100 million years ago. They are not geology. They are biology in geological disguise. The total biological contribution to the Earth's rock record is staggering when considered quantitatively. Approximately 50% of all sedimentary rocks by volume are carbonate rocks, limestones and dolomites that are of predominantly biological origin. The organic carbon content of the sedimentary record, the carbon from ancient biological material that was not oxidized but instead preserved in sediment and rock, represents the cumulative product of approximately 4 billion years of net biological carbon fixation. This organic carbon reservoir, distributed through the sedimentary record as kerogen, coal, oil, and natural gas, is estimated to contain approximately 10 to the power of 23 grams of carbon, an amount that dwarfs the carbon currently in the atmosphere, the ocean, and living organisms combined. The soil system that covers the land surfaces of the Earth is perhaps the most immediate and most directly visible example of biological planetary engineering. Soil is not ground-up rock. It is a biological system, a community of organisms so dense and so metabolically active that a teaspoon of productive soil contains more microorganisms than there are people on Earth. Soil is created by the interaction of rock with biology. Plant roots penetrate rock crevices and exert physical pressure that fractures mineral grains. Root exudates acidify the soil solution and drive chemical weathering. Fungi extend their hyphae through the mineral soil and dissolve nutrients from rock surfaces. Bacteria process organic matter and cycle nitrogen and phosphorus through the biological pool. The process of soil formation, pedogenesis, is fundamentally biological, and the soils that have developed over millions of years of biological activity on the land surface represent an irreplaceable biological asset, the platform on which all terrestrial agriculture and all terrestrial ecosystems depend. The research of the Biosphere 2 team, working with the Landscape Evolution Observatory, is demonstrating in real time what geological evidence shows across deep time. Life colonizing bare rock does not simply sit on top of the mineral substrate. It transforms the substrate, creating from sterile volcanic rock the complex biological material of soil. The organisms that pioneer this colonization, cyanobacteria, lichens, mosses, fix nitrogen from the atmosphere, dissolve minerals from the rock surface, and build up an organic layer that makes it possible for more complex organisms to follow. This is the process that has been operating on the bare rock exposed by glacial retreat, by volcanic eruption, by tectonic uplift, and by the original colonization of the continents, since plants first moved onto land 470 million years ago. It is happening today on the fresh lava fields of Hawaii, on the moraines behind retreating glaciers in Greenland and Antarctica, on the volcanic rocks of Iceland. And it is the same process that, at planetary scale over hundreds of millions of years, transformed the bare rock surface of the early Earth into the soil-covered continents that support the most complex and productive ecosystems the planet has ever produced. the nitrogen cycle. The system by which nitrogen moves between the atmosphere, the soil, the ocean, and living organisms is a biological creation. Atmospheric nitrogen gas, N2, is chemically inert under the conditions of Earth's surface, and the vast majority of organisms cannot use it directly. The nitrogen that living organisms require for building proteins, nucleic acids, and other essential biomolecules must be in fixed forms: ammonia, nitrate, organic nitrogen compounds. The conversion of atmospheric N2 into these biologically usable fixed forms is nitrogen fixation, and it is performed exclusively by certain bacteria and archaea. Without biological nitrogen fixation, the fixed nitrogen in the biosphere would gradually be oxidized and returned to the atmosphere as N2, and virtually all life on Earth would eventually starve for nitrogen. This is not a trivial observation. The nitrogen fixation that sustains the entire biosphere is carried out by organisms so small that they are invisible to the naked eye, operating in the soil, in the ocean, in the root nodules of legume plants, and in 100 other environments. The entire agricultural system that feeds 8 billion human beings depends ultimately on the nitrogen-fixing bacteria that convert atmospheric N2 into the forms that plants can use. The Haber-Bosch process, the industrial synthesis of ammonia from atmospheric nitrogen that was developed in the early 20th century, is the human-engineered equivalent of biological nitrogen fixation, and it currently produces approximately half of the fixed nitrogen used in global agriculture. Without either biological nitrogen fixation or the Haber-Bosch process, the global food system would collapse within years. The Earth system as it exists today is a co-production of geology and biology, and the biological contribution is not a superficial overlay on a geological substrate, but is woven into the fabric of the planet's chemistry at every level. The composition of the atmosphere, 21% oxygen, 78% nitrogen, trace amounts of CO2 and other gases, is a biological product. The oxygen was produced by photosynthesis. The nitrogen was modulated by nitrogen fixation and denitrification. The CO2 concentration reflects the balance between biological carbon fixation and respiration, modified by geological processes. If the biosphere was suddenly removed, if every organism on Earth died simultaneously, the chemistry of the atmosphere would begin to shift within decades, as the oxygen that is continuously being consumed by geological and chemical oxidation was no longer replenished by photosynthesis. Within millions of years, the atmosphere would be unrecognizable. The hypothesis that the Earth's biosphere operates as a self-regulating system, the Gaia hypothesis, first proposed by James Lovelock and Lynn Margulis in the 1970s, has been transformed by 50 years of subsequent research from a controversial metaphor into a serious scientific framework, though not in its original strong form. The strong Gaia hypothesis, the idea that the Earth is in some sense a single organism that actively maintains conditions suitable for life, has been criticized as teleological, implying purposeful behavior in an evolutionary context, where purpose is not a recognized mechanism. But the weak Gaia hypothesis, the empirical observation that the biosphere has historically maintained conditions within the range suitable for life despite external perturbations, and that biological feedbacks play a central role in this maintenance, is now well supported by the research reviewed in the Bristol and Exeter study, published in August 2025. The key in part of this research is that the Earth's biogeochemical cycles, the carbon cycle, the nitrogen cycle, the phosphorus cycle, the sulfur cycle, are not independent geological processes that life happens to participate in. They are biological creations, built up by the accumulated metabolic innovations of microbial evolution over 4 billion years. And they function as an integrated system that recycles the elements of life through the biosphere, the geosphere, the hydrosphere, and the atmosphere in ways that prevent the accumulation of biological waste products and the permanent loss of biologically essential elements to geological sinks. The modern biosphere contains approximately 700 billion tons of carbon in living organisms. The turnover time of this carbon, the time it takes for the entire carbon pool to cycle through the biosphere, is estimated at less than 8 years. In those 8 years, carbon is fixed from the atmosphere by photosynthesis, passes through food webs, is respired back to the atmosphere by decomposition and metabolism, is fixed again, and the cycle continues. This cycling of carbon through the biosphere at a rate of hundreds of billions of tons per year is the heartbeat of the Earth system. It maintains the atmospheric CO2 concentration. It drives the distribution of nutrients through the ocean and the soil. And it is the engine of the primary productivity on which all other life depends. We are one layer of this system, the most recent, and by geological standards, the most abruptly transformative. The numbers that follow make that transformation precise. The cyanobacteria that produced the Great Oxidation Event did not intend to poison the biosphere. They were doing what natural selection had designed them to do, capturing sunlight and using it to build organic molecules, releasing oxygen as a waste product. The organisms that were killed by that oxygen had no mechanism for responding collectively to the threat, no capacity for foresight, no ability to coordinate a response, no way to recognize what was happening at the planetary scale. The quantitative comparison between what the cyanobacteria did and what we are doing is both instructive and sobering. The Great Oxidation Event unfolded over approximately 300 million years, a geological timeframe so vast that the organisms driving it experienced no change in atmospheric oxygen chemistry within any individual lifetime or any individual species' evolutionary history. The rate of change in atmospheric oxygen during the Great Oxidation Event, measured in parts per million per year, was imperceptibly slow by the standards of any biological perception. What we are doing to the atmosphere of the Earth is happening at a rate that is, by the most conservative estimates, 100 times faster than the Great Oxidation Event, and probably closer to 10,000 times faster when compared to the most gradual portions of that ancient transition. The specific numbers are worth stating precisely. Before the Industrial Revolution, the concentration of CO2 in the atmosphere was approximately 280 parts per million, a concentration that had been relatively stable throughout the entire Holocene, the 10,000-year period of climatic stability in which all of human civilization developed. By 2024, that concentration had reached 422 parts per million, a 50% increase in approximately 200 years. The rate of increase is currently approximately 2 to 3 parts per million per year and accelerating. In the geological record, comparable increases in atmospheric CO2 concentration are associated with the major mass extinction events. The end Permian, where the Siberian Traps eruptions drove a similar-scale atmospheric perturbation, and the Paleocene-Eocene thermal maximum, where a sudden release of carbon from geological sources drove rapid warming and ocean acidification approximately 56 million years ago. The geological analogues for what we are doing are not pleasant ones. The ocean is the system most immediately affected by the rising CO2, and the effects are already measurable at global scale. Ocean pH has decreased by approximately 0.1 units since the pre-industrial period, a change that represents a 30% increase in ocean acidity because pH is a logarithmic scale. This rate of acidification is faster than anything in the geological record for the past 300 million years, except for the immediate aftermath of large bolide impacts. The organisms most vulnerable to ocean acidification are exactly the organisms that built the carbonate rock record of the past 500 million years. The corals, the coccolithophores, the foraminifera, the pteropods. The calcium carbonate shells and skeletons of these organisms dissolve as ocean pH falls below critical thresholds. And the coral reef systems that represent some of the most biologically diverse ecosystems on the planet are bleaching and dying at accelerating rates as ocean temperatures rise and pH falls simultaneously. The soil system, the biological infrastructure of terrestrial productivity that took 400 million years of biological activity to build, is being degraded at rates that have no geological precedent outside of mass extinction events. Approximately 1/3 of the world's topsoil has been degraded since the beginning of industrialized agriculture, driven by the combination of erosion, compaction, salinization, and the reduction of soil biological diversity through monoculture farming and pesticide application. Topsoil, the thin biological layer of 2 to 30 centimeters that supports virtually all terrestrial food production, forms at a rate of approximately 1 centimeter per 100 to 200 years under natural conditions. It is being lost at rates tens to hundreds of times faster than it forms. The biological infrastructure that the first land plants began building 400 million years ago, and that has been progressively elaborated by every terrestrial ecosystem since, is being dismantled by the agricultural practices of the past 2 centuries. The nitrogen cycle, the biological system that makes fixed nitrogen available for plant growth, is being disrupted at planetary scale by industrial nitrogen fixation. The Haber-Bosch process, which synthesizes ammonia from atmospheric nitrogen using hydrogen derived from natural gas, currently produces approximately 150 million tons of synthetic nitrogen fertilizer per year. This quantity of fixed nitrogen is approximately equal to the total amount of biological nitrogen fixation occurring in all natural ecosystems combined. The injection of this synthetic nitrogen into agricultural soils, rivers, and coastal waters has produced a global-scale perturbation of the nitrogen cycle that is manifesting as eutrophication of rivers and coastal waters, hypoxic dead zones in coastal oceans, and the emission of nitrous oxide, a greenhouse gas approximately 300 times more potent than CO2 from fertilized soils. The nitrogen cycle that microbial evolution spent 4 billion years building is being doubled in throughput in the space of one human lifetime. The biodiversity crisis that is currently underway, the sixth mass extinction, as it is now commonly called out, is in its biological and geological consequences comparable to the 5 previous mass extinctions in the geological record. The current rate of species extinction is estimated at 100 to 1,000 times the background extinction rate, the rate at which species would be going extinct in the absence of human activity, and is accelerating. The loss of species is not merely a conservation concern. It is a geological event with consequences for the Earth system that will persist in the rock record for millions of years, visible to future geologists as a sharp boundary in the diversity of fossil organisms, analogous to the boundaries that mark the end Permian and end Cretaceous extinctions. But there is an asymmetry between us and the cyanobacteria that is as important as the similarities. The cyanobacteria had no mechanism for understanding what they were doing. Their oxygen was a waste product of a metabolic process that natural selection had designed for individual reproductive success, and the planetary consequences of that waste product were simply invisible to any process operating at the biological scale. We have built instruments that measure atmospheric composition with parts per billion precision. We have developed climate models that can predict the consequences of specific emission pathways over century timescales with quantifiable uncertainty bounds. We have read the geological record of past climate transitions with enough resolution to know what CO2 concentrations of 400 parts per million produced in the Pliocene 3 million years ago, when the sea level was approximately 15 to 25 meters higher than today. We have, in other words, the equivalent of a planetary warning system that the cyanobacteria entirely lacked. The question this raises is not geological. It is philosophical and political. The Earth system does not care whether the organisms rewriting its chemistry are acting with awareness of what they are doing. The physics of the greenhouse effect operates identically whether the CO2 molecules were emitted by a conscious being or a bacterium. But the presence of awareness and the capacity for foresight changes the moral dimension of the planetary rewriting in a way that has no precedent in 4 billion years of biological history. For the first time, an organism can know that its metabolic activity is producing planetary-scale consequences, can model those consequences quantitatively, and can choose to act differently. Whether that capacity for choice will be exercised at the speed and scale that the pace of the current transition requires is the central question of the next century, a question that will be answered not by geology or biology alone, but by the political and economic systems that humans have built to coordinate collective action. The Earth has been rewritten before. It was rewritten by the methanogens that kept it warm under the faint young sun. It was rewritten by the cyanobacteria that poisoned the Archean biosphere and made complex life possible. It was rewritten by the first land plants that dissolved the continents and cooled the climate. It was rewritten by the carboniferous forests that stored their own bodies as coal. Every rewriting left the planet permanently changed, with winners and losers that could not be anticipated by the organisms doing the rewriting. The current rewriting is no different in its mechanism: metabolic activity producing planetary-scale chemical consequences, but it is different in one unprecedented way. The organism doing the rewriting knows what it is doing. What it does with that knowledge is the only genuinely open question in the 4-billion-year story of life and Earth. We do have these capacities. We are the first organisms in the history of the Earth that can understand the planetary consequences of their own metabolic activity, can model those consequences quantitatively, and can choose to act differently in response to what the models show. The Earth will survive what we are doing to it. It has survived the Huronian glaciation, the Great Oxidation Event, 5 mass extinctions, the Siberian traps, and the Chicxulub impact. It will survive the burning of the fossil fuels. The question is whether the conditions it maintains after the transition will be the conditions in which our civilization was built. The conditions of the Holocene, the stable, warm period of the past 10,000 years in which all of recorded human history has occurred. The biosphere has been rewriting the planet for 4 billion years. For the last 200 of those years, the rewriting has been ours to do. What we write next is not predetermined. But the geological record of the past 4 billion years suggests that the consequences of biological activity at planetary scale, intended or not, tend to be permanent. We do not live on a planet. We live inside a biological project, and for the first time in 4 billion years, the project is aware of itself.